Hi! So this is a video presentation on our primer that has been designed for polymerase chain reaction. We'll be creating a primer for the John Cunningham Virus DNA. The John Cunningham Virus, also known as the JC virus, is a common microorganism in which more than half of adults have already been exposed to. It usually resides in the urinary tract, bone marrow, tonsils, brain, and can be found in blood as well. The JC virus can lead to a disease called progressive multifocal leukoencephalopathy, in short, PML. PML is a serious brain infection in which myelin, which is the outer coating of neurons, are damaged progressively. This eventually leads to death of neurons in the brain. This also causes permanent disabilities and can even lead to death. There's also no cure for the JC virus or for PML.
The spread of the virus is unknown, but it is suspected that the JC virus may be picked up through the consumption of food or water containing the virus when young. JC virus remains latent in the human body in areas such as the kidney and is only reactivated when the immune system is weakened. The purpose of amplifying the gene would be to detect for the JC virus in the human body.
So now we'll be talking about the rules of designing primers and more about the criteria we took into consideration when designing our primers for the PCR of the JC virus. Rules of designing primers. The primer is recommended to be between 18 to 25 nucleotides in length and to have a GC content of 40% to 60%. The primer melting temperature is ideally to be 52 degree to 58 degree, and the maximum temperature should not exceed 65 degree. The melting temperature difference of custom primers should be not more than 5 degree between the pair of primers. Next is to avoid running 4 or more times of one base or dinucleotide repeats. For example, ACCC or ATATATAT, as it can cause primer mispriming. Also, avoid having primers which have secondary structures such as hairpin loops, dimers, and bulges.
These are our forward and reverse primers which have been drafted from Primer Blast. Our forward primer has a melting temperature of 57.7 degrees Celsius and GC percentage of 55%. It has two secondary structures, which are dimers and internal loops. They have melting temperatures of negative 171.3 and negative 119.9 degrees Celsius respectively. Due to the very low melting point, the secondary structures will not pose as an issue during PCR as they are hard to form. Our reverse primer has a melting temperature of 57.8 degrees Celsius and GC percentage of 50%. Similarly, it has one secondary structure, a dimer, and has a melting point of negative 108.6 degrees Celsius, which will also not form during PCR.
Now moving on to reason for detection. Patients that are undergoing procedures or have been recently diagnosed for illnesses that require the use of immunosuppressants or illness that cause immunocompromisation can undergo a test for the detection of the JC virus. This is because the virus can be reactivated to cause PML when the immune system is weakened. An example of such case would be optimizing HAART, which is highly active antiretroviral therapy, for HIV patients to prevent further deterioration of their immune system, or avoiding the use of immunosuppressants to prevent reactivation of the virus. This test of detecting the JC virus should also be conducted before prescribing patients with Tisabri, which is used to treat multiple sclerosis as it increases the risk of developing PML. So the materials and methods used for our project are Primer Blast and Gene Runner. We have come to the end of our presentation, and these are our references. Thank you for watching our video. Click here for more and click here to subscribe. Bye!